Hi, everyone. Welcome. My name is Sandy Cruz of Sandy K Nutrition, Health and Lifestyle Queen podcast. We are live here in Miami and having an amazing time. This is actually day 3, and I have the privilege of interviewing Christian Drapeau. He's a stem cell scientist, and I actually just got to hear him speak, and I'm so excited for this talk, you guys. And I'm going to let Christian just provide us with his background and what we're going to talk about today. Christian.
So my original background is neurophysiology from the Montreal Neurological Institute, McGill University, where I was working on memory and epilepsy. And then after I left, I was hired to work on specific plants, and the discoveries and the observations we did just led me into stem cell research. So I've been in stem cell research for the past 23, 25, 24 years.
You're too young for that, Christian. There's just no way. No. I'm fascinated with stem cells, and I've done, a little bit of reading, a little bit of research myself, but I think it would be important to begin the conversation to explain to people, what do stem cells do in the body?
Yeah, I think if I kind of frame a little bit your question, most people, when we talk about stem cells in today's world, it's a treatment. You go somewhere,
yes,
you get stem cells. So the question is, what kind of stem cells, what kind of injection? And what really is not talked about is that many of these stem cell treatments, the stem cells come from your bone marrow, your fat tissue, your blood. So that means they're in your body. They're not better because we take them out and put them back in. So for me, what I thought fascinating since day one in this whole field of stem cell research, which is if stem cells have such regenerative potential and they're already in our body, what is their role? What do they do? So in that context, a stem cell is a cell that has the ability to multiply endlessly and to transform into cells of other tissues. The best way to understand what they are is to define first what is not a stem cell. A stem cell, like a somatic cell, a cell of your heart. It will only beat, contract in its entire life. That will never do anything else, and it will never transform into anything else. So all the cells of our bodies are specialized. They do one thing, they will never transform. At the other end of the spectrum, you have stem cells. They are nothing. By nothing, I mean a stem cell does not have a function in the body other than its ability to transform into another cell that will become a somatic cells. So they are the mother cells of the body, the building blocks of the body. They are indeed the repair system of the body.
And every organ has stem cells. Like, how does that work? Because I have actually a couple of questions here. Maybe I'll start with how we've been taught stem cells. You take them out, you put them back in, or there's placenta stem cells that are taken from placenta. It gets very complicated. But the whole point is we know stem cells have regenerative capabilities.
Correct.
But it gets confusing because, like, as we get older, don't our stem cells just decline? Like,
stem cells declined both in quality and in number.
okay.
So this is just a reality that we have to face. The number of stem cells declines because the red marrow converts into yellow marrow very early in our lives. By age 30, we've lost about 90% of our red marrow. And that's why we discover in our 30s that, we're no longer Superman, Wonder Woman. It's that our ability to repair is declining significantly. So yes, there's a definite decline as we age, and it is probably the most fundamental core reason why we age and we lose our health as we age. The little caveat to this that I would add is that there is this understanding right now out there floating around. I've seen it in many interviews on YouTube and with people kind of echoing this statement that past 40 years old, your stem cells are worthless. And I think it's really an exaggeration that comes from a reality, meaning your stem cells are at 40 are not as good as when you were 2.
Right.
And at 60, they're not as good as when you were 40. But we have studies done, ongoing studies, where we see significant improvement, actually normalization of cardiac function in people with congestive heart failure simply by stimulating the release of people's own stem cells by taking plant extract. So I'm talking here 70 years old and above with chronic stable congestive heart failure. They release their own 70-year-old stem cells, and within 6 months, they have normal cardiac function. Just to say that, yes, stem cells are not as good as when you were 2, but to say that they are worthless would be almost like saying, at 50, are you as strong as when you were 15? No. Are you worthless? No.
No, I'm not.
No, you're not.
No, I'm not.
So your stem cells are good during your entire life. Not as good as when you were young, but they're good, and they do the job of repair. And that's why you age. You don't have as many, and they're not as good.
Okay, and if you're stimulating your own stem cells, wouldn't they be considered new? I don't know. Like, I'm kind of confused by that statement because I don't, as we get older, okay, the quality may not be the same, but if we're stimulating new stem cells, aren't they
Correct.
new?
They are new. They are new.
Right.
It's just the assumption in our understanding that when one cell divides into 2, that these 2 are identical to the first one. They are. In theory, they totally are. Now, take that division over the entire life of an individual. Now, these stem cells have telomerase in the bone marrow. So there's no attrition of the telomere. So they are immortal. They will multiply during the entire life of an individual. But there's over time an exhaustion, let's say, could be done because of a reduction maybe of mitochondria. I don't think the phenomenon has been studied well enough. Okay. I don't think we know why these stem cells are not as effective, but it comes from studies. For example, you take a 4-month-old mouse. So you take a mouse that will live, let's say, 16 months, roughly, okay? So you take now a 4 or 6-month-old mouse, and you trigger an injury, and you inject in that mouse stem cells extracted from a 2-days-old mouse, and in another group, stem cells coming from a 14-month-old mouse, geriatric mouse.
Okay.
And then you can see that the potential the boost in repair is much better with the young stem cells.
Okay.
So we can tell that stem cells that are younger work better in terms of repair, but they're still working even when they're old.
Yes. Okay. I get what you're saying now. That makes sense. So you're stimulating your own stem cells as opposed to extracting from placenta or whatever.
Correct.
And I think the most fascinating thing, since I heard your talk, is some of the studies that are coming out. And then we'll get into how this is actually done and the safety and any contraindications. So, maybe get into some of that, the studies. The studies are fascinating with the heart. And then I think you showed Parkinson's disease as well.
Yeah.
These are fascinating studies.
Well, a lot of the studies, because there's, let's say, 2 levels of studies. You have your clinical trials in humans, which do not allow you to have a mechanism of action. Like, I give a product to somebody that I know triggers stem cell release, but I have and then I have an improvement in these individuals. I cannot tell that it's the stem cell release that created the improvement. So these studies work, but in order to really document the mechanism of action, we need to go into animal studies.
Okay.
So there's really 2 levels into all that documentation. So in the animal studies, what has been clearly showed, and as you have seen in that presentation that I did, I'm choosing diseases or problems for which medicine, it's very well established, you don't recover from these. Yeah. You have diabetes, you cannot go somewhere where you're cured of your diabetes. No, you will get insulin for the rest of your life every day, and that's the treatment that medicine provides you. So if I can show that putting more stem cells in circulation can really improve diabetes, then I'm really showing something significant. So I've chosen those kinds of types of problem to illustrate really the power of your own stem cells.
Okay.
So with that in mind, you talk about the heart. Yes, studies in the heart where in mice model, they have shown very clearly that if you trigger heart attack and heart damage, heart attack, enlarged heart, scarring of the heart, fibrosis in the heart, and if you simply stimulate the animal's release of its own stem cells, we show a significant improvement, at times almost like a normalization of cardiac function.
Wow.
Just by putting more stem cells in circulation. So what we have done, so that is done with tracking stem cells and really documenting the benefits, showing images, slicing of the heart, histology of the heart, which you cannot do in a human.
Right,
So
right, of
once
course.
all of this is done, what we used is we use a plant-based stem cell mobilizer, so a blend of plant extract that you consume by mouth orally, which trigger the release of your own stem cells. That we documented very well. And then we monitored the benefit on the same condition that were studied in animals. So what we have documented a significant improvement in congestive heart failure, for example. People, what we just talked about with age, people that are, I think the age of the people we had in the study was from 68 to 74. So you're talking about people that we would consider, out of these statements that are floating around, that their stem cells are worthless.
Yes.
Every single one of the people, the participant in that study so far, have normal cardiac function 6 months later, just by releasing their own stem cells.
And it doesn't matter how old they are. And with the mice, I'm curious, like, were they young mice? Were they older mice in the studies?
Most of the time, I mean, I would have to go back in this study, but most of the time they do these studies in relatively young mice.
Oh, okay.
Because the cost of maintaining animals, and unless you want to really make a point that you're doing that in old mice, then my suspicion here is that it was probably, I would say, within 2 months old mice. The time to get them, let them being acclimated in the lab, and then you do a study.
Okay, so if we're doing this, does it matter? You're talking about plants, extracts that actually help your body to do this on its own.
Mm-hmm.
Depending on how old you are, would be dependent on how much you would need of these plant extracts, right? Or does it depend on the problem that you might be having? I know we can't say that it fixes or cures or heals any disease in humans, right? Technically. But, if you're trying, like, I'm 50, or I'm not 50, I'm going to be 54 in January. I still think I'm younger than 50, but if I want to have simply more energy and I don't have any actual issue, would I take a recommended dose? Could I overstimulate stem cell production in my body? Is that possible?
Okay. It's a very broad question. Let me approach it from the different angles.
Okay.
So the dose.
Yeah.
Is the dose dependent with age? For us as a dietary supplement company with limited budget to do that kind of research, to do a study to show what is the difference in the effect, whether somebody is 20 years old, or I have a group at 40, or I have a group at 60, and I have a group of people at 75, it may cost me $1 million to tell you.
I'll be in it for free. How about that? And I know you have costs, but I'm just kidding.
But so my point is that I would love to do that kind of studies.
Yeah.
What we did is that the people in our studies were between 20 and 70.
Okay.
This is the bracket of people, healthy people.
Right.
That's what we did in our study, to study the benefits of the product. So we quantified stem cells in these people. If I go back in those cases and I look at their age, yes, I can tell you there's a link between age and how many stem cells they have in circulation.
Okay.
When I normalize the data, meaning the percentage increased above their baseline level, there's no real difference with age. So if you start, let's say we say you can double the number of stem cells in circulation by taking 2 capsules of this blend of plant extract that we have. That means that average-wise at 50, you probably have about like 2 stem cells per microliter. So let's say we make that 4. If you are 30 years old, you may have 6 or 10. So you may have 20. At 5, you may have 100. So there's this sharp decline. But at any age, whatever additional I can put on. So let's say at 50 years old, you have about roughly 10 million stem cells in your bloodstream, roughly. So you take 2 capsules, you put in your circulation another 8 to 10 million stem cells. It's not a huge amount, but do that for every day for a month, you have put 300 million stem cells in your blood circulation. It gets pretty significant pretty quick.
What about for prevention?
So now for prevention, there is Because what is interesting here is that obviously medical research doesn't study health. It studies diseases.
Right.
Everything is about disease. So anything we draw as conclusion for health comes from studies on diseases. So as scientists study disease, so you typically take a mouse and a female mouse, you irradiate the female mouse, kills all the stem cells, you inject stem cells from a male mouse, they have the Y chromosome, so now you can follow these cells.
Okay.
So if you trigger, let's say, a stroke, whatever, you trigger a problem, an injury, a disease, and now you monitor the repair by triggering the release of the mouse own stem cells. And you can monitor the repair that is derived from stem cells by seeing the new cells in the tissue that have the Y chromosome, meaning they come from the stem cells. That's how these studies were done, to show how stem cells are the repair system of the body. And as they do these studies, what was realized, observed, many studies, is that while stem cells go where you have an injury, primarily in the background, they also go everywhere else. So there is a repair system and there is a maintenance renewal system. And out of those studies, then the question came out, is just, that's interesting in mice, but do we see that in humans as well? And there is one set of circumstances where you can study that in humans. And it's a woman that has leukemia and needs a treatment, which is irradiation or chemotherapy to kill all of her stem cells.
Yes.
And then receiving a bone marrow transplant from a male compatible donor. So from that day, that woman, all of the stem cells in her bone marrow have the Y chromosome. So any cell, stem cells going now to the liver, to the heart, whatever in the body, will have the Y chromosome. So they went into bank of tissue biopsies that came from autopsy. When one such woman that had bone marrow transplant from a male donor died, let's say 2 years, 5 years, 15 years, 20 years later. So now you look at not only the stretch of the tissues, but over time. And the conclusion is very clear. Now we realize every single organs of the body constantly is going through tissue turnover.
Yes.
But they all do it at a different rate. Like you say yes in the sense that it's obvious. Like it's obvious that the body goes through a renewal. But scientifically, it was not something that has ever been documented before stem cell research. It's an idea, but there was no data behind this. Now there is data. I can tell you, you have a new liver every 2-3 years, a new pancreas 4-6 years, half of a new heart every 25 years. In a world where we thought that the heart matures until about 8-10 years of age, and then that's what you have. If you lose cells, that's it. The heart cannot repair. Untrue. Now we know that the heart repairs as well with stem cells. So that means that, put disease aside, you're just healthy. You're still going through a process of tissue turnover. Now let's go back to what we were talking about before. By age 30 or at somewhere in your 30s, you cross that threshold where you don't have enough stem cells now to offset that normal cellular loss that is part of tissue turnover. So there's a point in your 30s when now your tissue begins to accumulate a daily deficit that will accumulate, and in 10, 15, 20, 25, 30 years down the road, it shows up as any one of those so-called age-related disease, which is a deficit that is more important in your pancreas, in that person's bone, in this other person's brain, this other person is the lung, the heart, whatever. So I proposed this concept of the stem cell understanding of disease formation in 2013. In a paper that was published in the journal, I think it was Stem Cell Research and Therapies. And at that time, I said, if that is true as an understanding, then there's one way to test it. Go and count the number of stem cells in people who have developed all those so-called age-related diseases, and count how many stem cells they have in their blood, and let's compare that with healthy people of the same age. Mm-hmm. We should see that they have fewer stem cells in circulation, and the disease is the result of this long-term, constant lower number, and therefore deficit in the ability to maintain the health of these tissues. So it shows up in the disease over time. So now many studies have been done. So you count the number of stem cells in people with atherosclerosis, erectile dysfunction, liver failure, heart disease, diabetes, emphysema, Parkinson, Alzheimer's, lupus, arthritis, COPD. The list keeps growing. They all have, in all those studies, less than 50% of what you find in healthy people.
Wow.
So that's my next book. Going through all the literature, all the studies, can we derive, because studies all count stem cells using a slightly different protocols. It's not easy you cannot just straight across take all these data and make calculations. So you need like an in-depth meta-analysis of that data. Can we derive a number of stem cells that we know under that number you will develop one of those age-related disease? That then makes stem cells a risk factor. The moment that it is a risk factor, we speak the language of medicine. Now you know that today, as a healthy person, you know that in 5-10 years, you are really at risk for developing whatever is the problem that, given your physiology, your biology, your genetics, your family, all of that. I know that you actually, honestly, if I were to say, what is your weak link in your physiology right now? You know.
Oh, I know my weak link.
We all know.
I know what it is. Yeah. And
that's where the problem will come if you don't have enough stem cells over the years.
And I do find it, the link between epigenetics and stem cells, right? So when do a lot of these genes start to express? Around midlife, for me, 30s, late 30s, thyroid cancer at 41.
Wow.
Right? So I'm just saying everything that you say makes a lot of sense. For me, I'm trying to put it all together in a context to say this would be a great modality for prevention of those genes to express.
The link between stem cells and epigenetics, at least as far as I'm concerned, as far as I know, is so young that there's very little that we can say.
Right. I'm thinking logically, right?
But your stem cells in your bone marrow are not exposed to the same thing that your tissues are exposed to. Not from a blood circulation, but I mean from a physiological standpoint. So can the epigenetics in your bone marrow be different than what you have in your tissues? Question mark. I don't know. So it's a very interesting question as to what is the link between stem cells and epigenetics.
Yes.
Because here's an example. It's not about genetics, but let's say telomeres, for example.
Okay.
Right now, there's a big discussion about telomeres and the importance of telomeres. Yep. However, the compounds known to so-called lengthen telomeres, when you consume these compounds as a human over a year's time, the benefits are very small. Better metabolic markers, better blood pressure to a small extent.
Those too.
It's very small.
Yeah.
So the data that is wow comes from my studies on mice that had a genetic manipulation to make them deficient in telomerase. So now when you reactivate telomerase, the rejuvenation is phenomenal. But that is not what happens in a human. So the result is fairly small.
Right.
And we measure that into lymphocytes that have a 3-month lifespan. What does it mean to measure telomerase or telomeres length in a population of cells that actually don't really live very long? We don't measure that in your heart, your liver, your tissues. So I'm not saying there's not something to that science. But what I'm thinking is that, let's say we talk about your liver or your heart.
Yes.
Or these tissues where telomeres length matter as an overall average length of telomeres in a tissue, of all the cells in the tissue. The longest telomeres are in your stem cells from your bone marrow because you have telomerase in your bone marrow. So that means a stem cells that migrates into a tissue, that stem cell that now becomes a liver cell is the cell in your liver, the liver cell that has the longest telomere. It just started its process. So flood your tissues with stem cells that now rejuvenate the tissue with new cells.
Yeah.
That tissue suddenly has, on average, a much longer telomere on average. So I do believe that if we focus on telomeres length, one of the most effective way is to flood your tissue with new stem cells by releasing stem cells from the bone marrow, and then you rejuvenate the whole tissue, and your average telomere length now has just lengthened.
That's amazing. I mean, there's some amazing stuff coming from this. So I have to ask now, because I think maybe I did ask before, but are there any contraindications to, stimulating your own stem cells?
I can take this question from various angles. Let me start by the one to me that is the most in our face.
Okay.
The plants that I have documented act as stem cell mobilizers. How did I discover them?
Yeah.
I just looked at Okay, so I discovered the first one that has an effect on stem cells, and I discovered it because that product was leading to benefits touching so many different aspects of human health. So once we documented that this was a stem cell mobilizer, I just asked the question, what else is known historically to be associated with many benefits? And let's go and test if it has an effect on stem cells. Medicinal mushroom, goji berry, all the adaptogens, those products that we know are associated with many things, but the main documentation that we have is, they're good for the immune system. Well, the immune system is just one part of your physiology. It won't link to like all the benefits to the heart and to everything else. And I tested them, and I find they all have an effect on stem cells. So it looks like their common denominator is an effect on stem cells. It appears novel because we've never looked at it, but it's turning out to be like very common actually. So I looked at things like medicinal mushroom, goji berries, sea buckthorn berry from the Tibetan plateau, used for 2,500 years in Tibetan medicine, Mongolian medicine, Chinese medicine for problems of the liver, of the heart, cardiovascular problem, diabetes. There's no data of safety, any issue of safety with sea buckthorn berry. No data of safety with fucoidan. I mean, there is actually data establishing the safety of these ingredients. So my point here is that I'm looking at products that all have a history of efficacy for many kinds of benefits and a long history of safe use. All I'm discovering is that during all these years, they were releasing stem cells we just did not know. And now the discovery says, okay, but if you release too many stem cells, can that be wrong for you? Well, it's not been wrong for 2,000 years. Why would it be wrong now that we just have the knowledge? So to me, at the onset, no, there's no issue. There's never been any issue with these plants.
Right.
Now, if we dive into the stem cell phenomenon itself, one common question is, can we exhaust our stem cells because we release them?
Yeah.
There's one study that to me is the answer to that question. And it comes from the question of, when you do cancer treatment and we take your own stem cells, we freeze them, and we give you back your own stem cells. So that's the process. So they will collect enough stem cells to re-inject 200,000 stem cells per kilogram of body weight. That's what they use to reconstitute your bone marrow. But when they isolate stem cells, in reality, they don't know how many stem cells. It's a very, very gross extraction. It's a very, very raw, crude extraction.
Are you also referring to, how when a baby's born, you take the umbilical cord, which has the stem cells, you preserve it. If that child ever got cancer, you can use those stem cells. That's like one method, right?
Yes, but now I'm talking about you having cancer.
Yeah.
Okay. So in order to, I give you chemotherapy.
Yeah.
It will kill your stem cells.
Right.
Okay. So before I trigger the release of your own stem cells with GCSF, the compounds that I was talking into all these studies in my presentation.
Right.
It's very well known. Everybody that has cancer treatment today, they trigger the release of their own stem cells. We harvest them from the blood, we freeze them, and after chemotherapy, we give you back your stem cells.
Yes. Okay.
So that's now a very common procedure.
Yes, it is.
To be successful, 200,000 stem cells per microliter, per kilogram of body weight, sorry.
Okay.
But the question is, this is a very raw, crude extract. I don't know how many stem cells I really have in this isolation. So the question was, if I have only one, but I can be sure that it's a stem cell, can one alone do the job? Interesting question. So they did a very sophisticated, I won't go into it because it's a little complex, but a brilliant protocol to identify one stem cells without tagging it with antibodies and markers, because then you affect it,
Right.
So they made sure that they had one cell, but it was a stem cell. And then they injected that into an irradiated mouse, and within 6 weeks, the entire blood is reconstituted, the entire bone marrow is reformed. If one stem cells can do it, and you have about 150 million stem cells that's in your bone marrow, and you release 10 million, 140 million remaining can do the job. And you don't actually have 150 million remaining, because the way that stem cells are released in the bone marrow is through a process called asymmetrical cellular division. When a mother cell divides into two daughter cells in nature, the DNA, there's one strand of DNA of the original DNA going into each daughter cells, and then a copy. They're identical daughter cells. It's a symmetrical division. In the bone marrow, it's asymmetrical division, meaning the original DNA going to one cell, the two copies go into the other cell, and that cell is released to go into the tissues. The original cell with the original DNA stays in the bone marrow so that your bone marrow retain your original DNA. Otherwise, you would have in your bone marrow a copy of the copy of the copy of the copy, and you would be 10 years old and you would be full of tumors and all kinds of mutations. So it's a powerful process to retain your original DNA in your bone marrow. So that means when you've just released 10 million stem cells, you're not lacking 10 million stem cells in your bone marrow. It stays constant. It has a powerful homeostasis. So there's no such thing as releasing too many stem cells.
Okay.
The person that is super healthy at 75, that has a lot of stem cells naturally, has been releasing more stem cells than the person that has fewer stem cells all his life. And the only outcome is healthier today.
Wow.
There's so many ways that we can look at that question. I understand where it's coming from, but you just step back and observe at life, and the answer is no. The more you release stem cells, the better off you are.
Mm-hmm. That's amazing. Now, let's get into these plants, what they are, and also efficacy. How do you know? Because I know in the presentation you mentioned that some will take more of STEMREGEN than others. And, how do you know? Like, does it
I don't.
Oh, okay.
I don't because age is one factor.
Okay.
Overall health is one factor. The quality of their blood circulation is one factor. These
I want to touch on.
These are all different factors that will show the efficacy, how much is needed for any one patient. So now I need to do a study. The cost of a study that is going to determine how much product you need depending of people, depending of their age and different condition. I mean, I don't have the $5 million that it would take to do that kind of studies. So we have to do some sort of generalization. All I can say is that after 20 years of doing this, all of our clinical trials is with 2 capsules, 3 times a day.
You mentioned the quality of the delivery system. How, because you also in the presentation talked about the capillaries and how is this actually delivered into the body? Because that matters. You also mentioned nattokinase, which I'm a huge fan of.
I believe that with all the science now that is coming up with nitric oxide, it's bringing some focus on microcirculation. But I think it is still a completely under-considered reality of our physiology. I take a blood sample into a highway, which is your vein, I'm into a 8 lanes highway, and I take a blood sample, and by determining what is in that blood, I assume what is happening in your body, and in reality, what you have in your blood only means something if it can reach the very fine capillaries where exchange is taking place. Otherwise, it doesn't matter. It's like having tons of money into the banking system, but you can't withdraw that money. What good is it for you? Mm-hmm. So capillary circulation is essential for nutrition, delivery of minerals and everything, and delivery of cells, immune cells and stem cells. Stem cells are big. They're not all big. You have V cells that are very small, but your typical mesenchymal stem cells can go as high in the body. I mean, when you take them from umbilical cord, adipose tissue, they tend to be larger. The one you release from your bone marrow are smaller, but many of them are bigger than a capillary. Let's say 20 microns. Your capillary is 8 to 12.
Okay.
So they're bigger. So that means they need to squeeze themselves. They're not rigid. So they need to squeeze themselves. So that means you need good lipid profile in your body. Polyunsaturated fatty acid, DHA, PEA, all of those change the fluidity of your membrane. Plasmalogen. These are good compounds that will affect the health, the flexibility of your membrane, essential for the ability of cells to squeeze themselves and find capillaries. You need your capillaries to be able to dilate. That means nitric oxide, an effect on the smooth muscles of capillaries, but also the ability of the capillary themselves to distend. And that is a consequence of the surrounding of these capillaries. A capillary is not like a vein or an artery. You cannot dissect a capillary. A capillary is a tunnel into your connective tissue that is covered with epithelial cells. So it's completely dependent on the health of your collagen that is everywhere in your body. So it's collagen formation, elastin formation, all the connective tissue. So that is the health that you need to support for proper delivery of stem cells. So that's why, and then hemodynamics. If your blood is too thick, because you have rouleau formations and that kind of thing, you won't have delivery of cells in the smile capillaries.
Right.
You've got a rouleau formation at the entrance of a capillary, your stem cells will just bounce off of that and will continue. That area of your body is deprived of the ability to repair.
These are capsules. What about the gut? The condition of the gut. Does that matter?
I have never studied the impact of the condition of the gut on these plants. And the reason why I have not done it is because as a researcher, you need to follow the scientific method.
Yeah.
As a researcher, I give the product, it works. Then
Yes.
why do we have to worry about what's happening in the gut?
I
it
get
works.
it. Yeah.
So it's never a question that came up. If it worked in half the people and didn't work in the other half, then I would have said, why is it not working?
Right.
Then I might have gone in that direction.
That
So
makes
right
sense.
now, everything that I have in terms of data is that it's independent from gut conditions.
Okay, let's talk. Can we talk about some of the conditions?
Sure.
Are we? Because
Let's not mention product brand, and then we can
Okay.
talk about
Okay.
about
Because, I'm just curious. There's some conditions that are very common, like arthritis as we get older, things like that.
arthritis. Clear demonstration in the scientific literature. If you inject stem cells, increase the number of stem cells in circulation, you can have significant benefits. We have had one case that we have published actually in the scientific literature many years ago of somebody who was using one of the first plant that I documented acting as a stem cell mobilizer. And she started with deformed ends and feet, so 18 years of evolution. So it was very deformed. She needed assistance to bathe, to eat, to dress. She was not walking. And then she started to take the product, and about 2-3 months later, she started to really have more flexibility. Another month or two, she no longer needed assistance to bathe herself, to eat. She started to walk with a walker. So fast forward a year later, she was walking unassisted, living unassisted. She still had arthritis, but the improvement in her functionality was enormous. She misread the label. She took 1 capsule a day. So you see, when we go back into the dose, we have people who have amazing results with very little, less than what we consider a dose. I did the science on 2 capsules. 2 capsules contain the cumulative quantity of the plants that have been shown to have the effect on stem cells. I have just enough of the sea buckthorn berry extract, of the fucoidan, of the allomacroclad, that kind of stuff that we have documenting having an effect on stem cells. So I can only talk about the effect of 2 capsules. I have not studied the effect of 1 capsule. So I can't talk about it. But can people use it? Of course. But in my study, we do 2 capsules 3 times a day for simply the reason that how foolish would I feel 6 months down the road concluding that I didn't see the result that I was expected, maybe I just did not give enough. I've used up time from a clinic, commitment from a clinic, patience, cost. So just to maximize the results, maximize the study at a time when we want to demonstrate that endogenous stem cell mobilization, the release of your own stem cells, can have a significant impact on health. That's my first aim right now.
Yeah.
So I can look at those later on. Right now, I want to demonstrate that part so people let it sink in. Yes, your stem cells can make a huge difference for you. Then we can go into other consideration.
You also mentioned diabetes, type
Yep.
2 diabetes. I think we talked about MS as well, didn't we?
Yeah.
Like, there's a lot of these debilitating diseases that people encounter as we age. And if we simply increase the number of stem cells, here's a question for you. I know the answer because I cheated. But how do your stem cells know where to go? How do they know?
Yeah. It's an interesting question because it's sometimes either part of I would say, a beautiful thinking, but it's just like to say, well, your body has an intelligence. Or it goes on the other end, which is it's nonsense, that stem cells would do that, because how do they know where to go? And it's just to bring it down to just how the body works. Let me put it this way. Let me start by answering, if you cut yourself and you have a bacterial infection, how does your immune system know that this is where the immune cells need to go to kill the bacteria? It's a very well-known process in the body.
Yes.
Your immune cells there will detect the presence of bacteria, will take off into your lymphatic fluid, will be captured by your lymph node. In your lymph node, will talk to the immune cells, other immune cells, show them that there's a bacteria there somewhere in your body. Then a bunch of immune cells are released. They go everywhere, but in that area, your immune cells are releasing signals that attract the other immune cells. When they go in that area, they're migrating in that area, and they're attracted to the bacteria, and they kill the bacteria. It's a well-documented process.
Yes.
We call it the immune system. Now we have an organ, the bone marrow, that releases a cell called the stem cell, CD34 cell. When you have an injury, the area of the injury releases one specific compound called stromal-derived factor 1. It's released locally in an area, so it goes into the capillaries, the fine vasculature of the area affected. So when a stem cell goes in the fine capillaries and suddenly that molecule is there, stem cells have a receptor for that molecule, specific. When that molecule touches the receptor, the stem cells expresses adhesion molecule and cling to the capillary. So they stop. The blood continues, but they stop. Now the receptor is still there. If I keep sensing that molecule, that means there's a real injury from there. So now the stem cells will secrete enzymes that will digest the attachment between two capillaries, so a hole in the capillary. So now the stem cell will secrete another generation of adhesion molecule that will make the stem cells migrate, throw a part of itself through the hole, and then will slowly pass itself completely through the hole until it has completely migrated its content on the other side of the capillary wall. So now you're into connective tissue of that organ. Stem cells will secrete another adhesion molecule that connects to hyaluronic acid in that tissue. And the stem cell will crawl following the gradient, the concentration gradient of SDF1. That's how your stem cells know how to detect a tissue that has an injury, migrate in it, and then walk to the site of the injury to repair where the injury is. It is your repair system. Just like you have an immune system, you have a repair system.
That's fascinating.
Isn't it?
It is so fascinating, and you're so passionate. I love it. Obviously, your answer was way more succinct than what I would have said from what I learned from the lecture this morning. But that's amazing. I did read your book. What's the name of your book, Christian?
Cracking the Stem Cell Code.
Fascinating. And I do feel that even although I do have a little bit of science, nothing close to your training, but I think anybody could understand it.
I wrote it. Okay, it was the first book ever published about the natural role of stem cells in the body. So as a first book with something that is that scientific, if you want, if I'm talking and I'm saying, well, we have a new system, keep in mind, the last system was in 1906 or early 1900s with the immune system, the endocrine system. So a century later, we say we have a new system in the body. I mean, it's presumptuous, you know? Yeah. So it needs to be very well documented. So I wrote it to answer all scientific questions. But as I wrote in the preface of the book, I said, but my aim is for the general population to be able to understand. So at any time, if the compromise is precision versus comprehension, I'm going to err on the side of comprehension. So there's tons of science in it, but I try to write it as much as possible as the story of your body.
Yeah, I think you have to appeal to anyone. If you want to appeal to the masses and explain how it actually all works, you've got to appeal to everybody, right? Okay, this has been an amazing conversation. I would love for you to let everybody know how they can find out more, how they can find out anything about you, Christian.
Our website, STEMREGEN.co, so they can see the product that we have, but also there are tabs there on all the research and everything is listed on the website. And I put tons of information on either Instagram, TikTok at Stem Cell Christian. If I say Christian Drapeau, most people can spell my last name. So
I can.
Because
I'm
you're
Canadian.
part French.
Yes, I understand. And I have to say, your TikTok is so well done. You guys have to go and follow him. Thank you so much, Christian. It's just such a pleasure to talk to you, and I'm so honored to meet you.
My pleasure. Thank you so much.
Thank you.